Bismillahirrahmanirrahim. This is the sixth video in the series of molecular bases of the cancer. In this video, we will discuss the cancer cell metabolism. These are the references for this video. Every cell utilizes glucose to generate energy, and this energy is used to form and repair DNA, RNA, lipids and protein. The amount of nutrient which is used by this cell depends on its function. Glucose enters the cell via its transporter. The main is GLUT1, while GLUT2, 3 and 4 are also useful. Aerobic glycolysis is a type of glycolysis in which glucose through enzymatic reaction is converted into pyruvate and two net ATPs are formed. The pyruvate plays a central role in metabolism of normal and malignant cell. It enters mitochondria, and by the action of pyruvate dehydrogenase it is converted into acetyl-CoA. This pyruvate may enter Krebs cycle in two ways: after conversion to oxaloacetate or after conversion to acetyl-CoA. Ribose 5-phosphate is formed by glucose. As glucose enters into the cell, it is converted into glucose 6-phosphate, then into ribose 5-phosphate that is incorporated into nucleic acid. Acetyl-CoA is formed from the pyruvate, which when enters into the mitochondria by the action of pyruvate dehydrogenase it is converted into acetyl-CoA, and entry into the Krebs cycle it yields 36 ATPs. Acetyl-CoA can also exit mitochondria and responsible for lipid biosynthesis. The amino acids are also synthesized, as the amino acid membrane transporters are responsible for the entry of the essential amino acid which cannot be synthesized by the body or cell. They enter through the membrane transporters, and other non-essential amino acids are synthesized by the cell from pyruvates or its metabolites. Glucose uptake can be due to exogenous signal and endogenous mediators. The exogenous signals are insulin and insulin-like growth factor 1 that binds with its receptor and causes the intracellular signaling that is responsible for further effect. The endogenous mediators are AKT-mTOR pathway that is responsible for protection from apoptosis and stimulation to proliferate by various proteins. The major effector of AKT is mTOR protein that is responsible for the uptake of amino acid and production of amino acid transporter also, and it is also responsible for increased lipid and protein synthesis. This AKT is blocked by phosphatase and tensin homologue, that is P10, and insulin-like growth factor 1 also causes the release of increased concentration of MYC protein that are responsible for expression of GLUT1, and this increased GLUT1 is responsible for import of glucose. So how this metabolic activation affects cancer cell metabolism? When insulin-like growth factor 1 or insulin binds with its receptor, the signaling activation occurs, and main signaling pathway is AKT, and one effector of AKT is mTOR, while AKT is also responsible for increased survival and proliferation, and it also is responsible for increased nutrients to the cell. The mTOR is the key effector of this AKT, and it increases the amino acid transporter entry, and as a result entry of the amino acid is increased and increased proteins are formed within the cell. Then mTOR also responsible for increased formation of lipid. It work with KRAS and MEK and increases the GLUT1 transporters, and as a result the glucose is entered more in the cell. It is responsible for increased energy generation and DNA formation. A special characteristic of metabolism in cancer cell was studied by Otto Warburg in 1930, and this characteristic is termed as Warburg effect. The cell ferment glucose to lactic acid using glycolysis even in the presence of oxygen. This is termed as Warburg effect. What is that? As in normal cell, when the glucose enters into the cell, it is converted into pyruvate, gives 2 ATP, and then this pyruvate enters into TCA and gives numerous ATPs. About 36 ATPs are formed in the normal cell. But what happens in a malignant cell? There is this is under Warburg effect. The glucose enter into the cell, and instead of the pyruvate is formed and it gives 2 ATP, and instead of going into the TCA or Krebs cycle, this pyruvate goes to forms lactate, and only 2 ATPs are formed by the formation of pyruvate. This is in contrast to increased demand of the ATP or increased demand of energy for this rapidly proliferating cell, as it is under the stress of proliferative stress. Then this pyruvate is converted into lactate by the lactate dehydrogenase, while the pyruvate is converted into the acetyl CoA by pyruvate dehydrogenase. The monocarbohydrate channels are also formed, and these are responsible for the export and import of the lactate. The lactate can come from the neighboring tumor cells into the cell, and it can go from this cell to the other cells to help in the proliferative activity. This pyruvate is responsible. This lactate is also converted into the pyruvate, and this pyruvate is responsible for the proliferation, for the formation of the macromolecule such as protein, lipid, and other macromolecular synthesis to cope up with the rapid proliferative activity and to form the building blocks rapidly. Another source of this is acetate. In this, the acetate is converted into acetyl CoA, and this acetyl CoA then enters into the Krebs cycle or TCA cycle and responsible for lipid synthesis. Glutamine is another source that is converted into alpha ketoglutarate and then is responsible for cellular metabolism. The exogenous insulin intake will result in more glucose inside the cell. So this explains the obesity with high circulating level of insulins and its association with cancer. The increased formation of GLUT1 is under the influence of oncoproteins such as BRAF, KRAS, and c-MYC, and this c-MYC is also responsible for increased glutamine transport into the cell and upregulation of the lactate dehydrogenase. This lactate dehydrogenase is responsible for the interconversion of pyruvate and lactate. The MYC dependent tumors are often inhibited if LDH or glutamine is decreased. Hypoxia inducible factor is also responsible for increased expression of GLUT1, and this hypoxia inducible factor is expressed in decreased oxygen concentration, or if von Hippel-Lindau gene is lost, or even in the absence of hypoxia, if there is mTOR activation, this hypoxia inducible factor will be present, will increase. This is also responsible for glutamine transport into the cell, and it also favors glycolysis as it blocks pyruvate dehydrogenase. For the description of this Warburg effect, the Otto Warburg received Nobel Prize for 1931. So the cancer cell is in constant hunger for glucose, and this increased glucose is converted into lactate through aerobic glycolysis, and this glucose hunger is clinically visualized by positron emission tomography scanning, that is PET scanning. In this technique, the 18-fluorodeoxyglucose is injected into the body. This chemical is non-metabolizable derivative of glucose, and it is preferentially taken up by the tumor cells and also by normal actively dividing cells such as bone marrow cells. The tumor suppressors try to regulate the cell metabolism. The VHL prevent hypoxia-inducible factor alpha from redirecting the cell energy production toward glycolysis. It has an E3 ubiquitin ligase component that results in polyubiquitination and degradation of hypoxia-inducible factor. P10 as P10 activity, as the activation of mTOR will result in enhanced glycolysis, that is the result of AKT triggering through phosphatidylinositol 3 kinase, and phosphatidylinositol 3 kinase is blocked by P10, that is phosphatase and tensine homologue. So P10 decreases mTOR activity, reduces hypoxia-inducible factor, and limit GLUT1 production, as hypoxia-inducible factor was responsible for increased expression of GLUT1. So GLUT1 is decreased by the increased activity of P10. Also, it inhibits hypoxia-inducible factor directly. So this P10 also prevent hypoxia-inducible factor alpha induced blockage of mitochondrial use of pyruvate to drive the TCA cycle forward. So P10 is a controller of the cellular metabolism, and it suppresses the metabolic changes that power the cell cycle activity. This mTOR has got numerous activities such as increase hypoxia-inducible factor expression, increase amino acid transport, decrease TCA cycle activity, and increase glycolysis. But when P10 blocks the or dephosphorylates the phosphatidylinositol 3 kinase, it will result in cessation of all activities downstream that makes it impossible for the cancer cell to generate the building blocks for the proliferation. Besides its tumor suppressor effect, P53 is also associated with the regulation of the cellular metabolism. The tumor cells that have lost P53 have been associated or shown the Warburg effect. P53 is activated by AMP kinases in response to metabolic stress, and there is upregulation of trigger, that is TP53 induced glycolysis regulator, that will result in block of aerobic glycolysis and shunts its intermediates to other pathways. The decreased glucose transporter, that is GLUT1 synthesis, will result in impeding of the glucose entry into the cell. It blocks the nuclear factor kappa B, thus it hinders its direct and indirect activation of glycolysis. The nuclear factor kappa B also upregulates hypoxia-inducible factor alpha 1. increased synthesis of a stimulator of cytochrome oxidase SCO2 that will result in increased mitochondrial electron transport that will increase pyruvate and glutamine importation into the mitochondria and incorporation into TCA cycle by upregulation of pyruvate dehydrogenase. Decrease MYC level by activating microRNA 145, that is the direct inhibitor of c-MYC, will result in decreased c-MYC stimulated hypoxia inducible factor production and increased oxidative phosphorylation. P53 indirectly impedes fatty acid biosynthesis, and it also upregulates intermediate molecules that can trigger autophagy. The net effect of the P53 is to shunt the energy production away from the glycolysis and toward oxidative phosphorylation. Hence, the loss of P53 it leads to Warburg phenomenon. P53 related metabolic protection is triggered by a series of events in which AMP kinase is activated, and this activated AMP K inhibits mTOR, and pharmacological stimulation of AMP kinase has been used in tumor therapy, and the pharmacological agent is usually metformin. This picture is showing the regulation of cellular metabolism by P53. Besides its other function, P53 is a critical metabolic regulator. It is activated by increased metabolic stress that will result in increased adenosine monophosphate protein kinase or AMP kinase that will activate P53. P53 directly downregulates GLUT1, hence it decreases glycolysis. Also, it downregulates nuclear factor kappa B, that is also responsible for decreased glycolysis, and it is also responsible for increased expression of TIGR, that is TP53 induced glycolysis regulator, that is also another factor for the decreased glycolysis. So, all these three, the decreased GLUT, decreased nuclear factor kappa B and increased TIGR, are responsible for decreased glycolysis. Then it increases TCA cycle activity as P53 upregulates SCO2, that is a stimulator of cytochrome oxidase that increases mitochondrial electron transport. It also increases pyruvate and glutamine incorporation into the tricarboxylic acid TCA cycle by upregulation of the pyruvate dehydrogenase. It also upregulates microRNA 145, which is responsible for downregulation of c-MYC, that also result in prevention of c-MYC mediated metabolic effect. Then P53 downregulates key enzymes that mediate fatty acid synthesis. Hence, by this mechanism, it prevents cancer cells from achieving their malignant potentials. The isocitrate dehydrogenase is an enzyme of TCA cycle, and it is a potent tumor suppressor. It is mutated in many malignancies such as malignant gliomas, myelodysplastic tumor, cholangiocarcinomas and sarcomas. The gain of function alteration will result in a different production of the protein, that is R2 hydroxyglutarate, and this will inhibit the TET family of DNA hydroxylases, and it also inhibit family of histone demethylases. So 5-methyl cytosine hydroxylation and TET2 related histone demethylation are not available to undo the downregulation of the tumor suppressor gene promoter by CpG and histone methylation. This inactivation of TET tumor suppressor facilitates tumor development. The Warburg effect describes the tumor cell metabolism, but tumor cells are not living in isolation. They have got their neighbors that are the stromal cells. That is the important component of the tumor, the stroma. The tumor cell metabolism create an environment of increased reactive oxygen species. So what is the mechanism of the action of these tumor cell derived signals on the stromal cells? The tumor cells secrete reactive oxygen species that act on the stromal fibroblast, and there is increased secretion of nitric oxide from the fibroblast that will lead to the mitochondrial injury. As a result of mitochondrial injury, there will be further increase of reactive oxygen species that will be taken up by the tumor cells, and it will result in increased genomic instability in the tumor cells. The increased concentration of reactive oxygen species is further worsen the mitochondrial injury and may result in mitophagy. So when we see the holistic picture of the series of events, the tumor associated fibroblast undergo mitophagy. As this damaged mitochondrion is taken up or is eaten up by the cell itself, so it will increase the aerobic glycolysis, and increase aerobic glycolysis in turn will increase the lactate concentration in that environment that will be taken up by the tumor cell through monocarbohydrate transporters, and when these lactate are taken up by the tumor cells, they are used for the formation of energy, and also as a substrate for the macromolecular synthesis. So how autophagy is responsible or is disturbed in cancer cell? The autophagy is a process of recycling and removing mostly of the cellular constituents, damaged cellular constituents, and it is basically a cellular response to supply metabolic needs in the time of stress, such as starvation. This autophagy is accomplished by BACLIN1 that is mutated in most of the cancers. And if it is mutated, the autophagy is not working properly. The autophagy mechanism is not working properly. That will accumulate the oxidant injury. And this accumulation of oxidant injury will start a vicious circle of further oxidant injury and accumulation and aggregation of reactive oxygen species within the cell. The P62 is central to tumorigenesis, and it is overexpressed in many of the tumors. Without the P62, the oxidized aggregate do not form, and the resultant genetic damage, which is due to these reactive oxidant species, does not occur. This accumulation will lead to increased rate of DNA mutation that will be responsible for genomic instability. The other mechanism by this failure of autophagy is cell death. As there is no intact p53, so cell is not going to apoptosis pathway. In fact, it goes to necrotic pathway, and this necrosis of the cell will invite another actor in this on this stage of tumor, that is tumor associated macrophage, that will facilitate further tumorigenesis. The tumor suppressors such as P10 and tuberous sclerosis protein facilitate autophagy, while AKT, BCL2 and mTOR, that are the oncogenic stimuli, they impair the process of autophagy. The role of p53 is ambiguous, and as in some instances it promotes autophagy and in others it impairs autophagy. The BACLIN1 is the gene that is involved in the process of autophagy, and it is mutated in most of the cancer. The antineoplastic medications are also induce process of autophagy in tumor cells. The quiescent cell is dependent on Krebs cycle for the generation of energy, and if there is deficiency of the nutrients, it activates autophagy, the process of self-eating of its own organelle. While in the growing cell, which may be a normal cell or a tumor cell, there is increased expression of glucose transporters that are activated due to action of the AKT. This AKT is under the influence of oncogene, for example phosphatidyl inositol 3 kinase, and due to increased expression of there is glucose transporter, there is increased glycolytic intermediate formation that under the or in the presence of pyruvate kinase will lead to formation of the macromolecules. There is also increased formation of lactate, and alternatively increased glutamine is also utilized. In cancer, the oncogenic mutation involving growth signaling pathway and other key factors such as MYC deregulate these metabolic pathways.